Yeah, I know Christina as one of our own health freedom advocates, absolutely, and she definitely defines the term in the sense that she dedicates her professional life to lobbying elected officials, staying on top of all the myriad laws and statutes and craziness that they're putting us through, and she's a wealth of knowledge. So I'm really excited to hear what she has in store for us tonight. The presentations on PCR tests, the mask wearing, all the crazy interventions during this pandemic. But she's just not going to rehearse things that you already know, many of you. She's going to prepare you to be able to protect yourself and then to advocate for change. So Christina, go ahead and say hi.
Hi, so my name's Christina Hildebrand. I am the founder and president of A Voice for Choice Advocacy. We educate and advocate for informed choice and transparency of what goes into your body, be it food, air, pharmaceuticals. We basically want you to know what's in the stuff, and then for you to choose whether you take it into you or not. And so I'm going to speak tonight on PCR testing and vaccines. But my background, my career is in market research. So I do market research for various Silicon Valley companies. I also have a food delivery service, which I'll talk a little bit about tomorrow in another talk that I'm doing called Real Food Bay Area. We deliver organic, nutrient-dense, Western priced food throughout the Bay Area. And then I have A Voice for Choice Advocacy, which is our 501c4, and our A Voice for Choice, which is our 501c3. And they basically do the same thing. One educates and one does advocacy work. So tonight's talk, I just wanted to talk on PCR testing and COVID vaccines. So part of what we do in our whole educating and advocating for transparency is this last 2 years or last year has really gotten into COVID. And I just wanted to share, as I said, the organization, we want you to have the choice. So for me to say you should not vaccinate is as bad as me saying that you should vaccinate, in my opinion. I have my personal opinions on vaccines and whether I would vaccinate myself, but every single person needs to make their own decision on that. The same with PCR testing. You need to know what it is. You need to know how to use it. You need to know when it's useful and when it's not useful. The same thing with vaccines, the same thing with anything in life. And so I wanted to kind of go through the first half of this talk is on PCR testing. The second half is on the COVID vaccine, and then it's kind of solutions of where you can go, what you can do, and what our life looks like in the future. So where are we at today? I said last night as we were doing introductions, I truly, if you have not read A Brave New World in 1984 in the last year or two, many people read them in school, I would say go read them, because I do think we're at a combining of the two at the moment. And the question is, does life become as they are portrayed in those, or do we do something about it? The portrayal of those two really is what has been talked about at the World Economic Forum. If you go onto the World Economic Forum's website, if you haven't done so, you really should, because they lay it out in black and white what they want the world to look like. And COVID is a part of that. It is absolutely a part of that. And how the dynamics of where we go from here, the new normal. This week or last week, they had the Davos agenda, the Great Reset meeting, that basically, if you listen to their speeches, it lays out exactly how the corporations of this world want the world to look like. And I'm a big believer you have to listen. You can live in your own bubble, but I truly look at both sides. So when I look at the vaccine issue, I'm on as many pro-vaccine groups as I am on quote unquote anti-vaccine and pro-vaccine groups, because I want to see what the other side is saying and what the other side is, what is their truth, because it's truly what the truth is for you. I have my truth, and that's different from everyone else's truth because of the amount of reading and research that I've done and the people I communicate with. And so you need to work out where your truth is. So going into PCR testing, as I said, I'm going to first talk about PCR testing, and then I'm going to get into the vaccines. So what is just COVID testing in general? There are 4 types of COVID tests that generally fall into 4 types. There are many more types if you go down sort of subtypes. But there's the PCR test, which is the polymerase chain reaction tests, antigen tests, which are quick turnaround tests, antibody, which is serological tests, blood tests, and then T-cell tests, which are also serological tests. Just to be clear, none of these tests for COVID, for SARS-CoV-2, which is the virus that they test for, SARS-CoV-2 is the virus, COVID-19 is the symptoms and the disease. It's like HIV and AIDS. So they're testing for SARS-CoV-2. None of these have been fully FDA approved. So none of them have gone through the approval process. They've just been put on the market. They meet certain criteria, which is a very, very low grade criteria. If they've been used, they've been approved for or authorized for emergency use. So these are not tests like other tests. So if you go in and get, for example, a measles titer test, which tests whether you have antibodies to measles, that's been fully FDA approved. It's gone through all the clinical testing that it required to get FDA approved. These have not had that at all. So I'm going to go into the PCR testing specifically because that is the one that is most used. Let me just go back to this. The PCR and the antigen tests are to test whether you have the disease currently. The antibody and the T-cell tests are if you had the disease, if you had the virus and you created some sort of immunity to it, those would test for that. So they are post getting COVID-19, and I know people question whether there is such a thing, but if you had the virus and you created antibodies, you would go with the antibody or the T-cell test. The polymerase reaction and the antigen tests are the two that if you have symptoms, you would be testing for it, or in this case, for the first time ever in our history, we're testing asymptomatic people as well. So looking at the PCR test, so the PCR test basically searches for the nucleic acid from the SARS-CoV-2 virus that's been identified. And this is the most accurate way to tell if an individual has the virus. And I use the word most because it's not a very high bar to get to most. There are no other tests that really test for it. The only other one that you could use is an ELISA test or an ELISA test, but those are incredibly expensive tests, and so you're not going to be doing that on the masses. Interestingly, PCR tests are not technically designed to detect contagious viruses. So Kary Mullis, who created the PCR test, got a Nobel Prize in chemistry for doing that. He specifically said that it is not good to be used for contagious viruses, but yet here we are using it because it is the most accurate way to tell if an individual has the virus. I'm going to go into how a PCR test works and what it does on the next slide, but just to go into it, there are more than 300 PCR COVID tests that have received emergency use authorization from the FDA. And are being used to identify patients with the virus. As I said, none of them have been fully approved. If you read the emergency use authorization sheets that come with it, most of these PCR tests, the large majority, have been approved for patients with symptoms or if someone has been exposed and believes they have the disease. So that's what the emergency use authorization is approved for. Had a big back and forth with the FDA because the FDA allows off-label use of products. So if you have a drug, you can use it, a doctor can use it off-label if they think it's appropriate. Usually for emergency use authorization, off-label use is not allowed, and it specifically says that. So for example, for the COVID vaccines, which are also emergency use authorized, they are only allowed to be used for that purpose as a COVID vaccine. The testing, interestingly enough, they didn't put that may only be used for people who actually have symptoms of COVID. So they are allowing off-label use, which is why it's allowed to be used for asymptomatic people, even though none of them have been authorized for asymptomatic people, or I shouldn't say none, there are a few that have been authorized, but not very many. So this is how the PCR test works. It goes through various steps. This is kind of chemistry 101, probably a bit more than 101. I'm going to go through it. If you don't follow it, I'll give you the general of what this actually means. But the first piece is you take a sample. So right now that's either a nasal swab or a saliva test. It's heated to separate out the strands of DNA through a thermocycler. It denatures the DNA. It then attaches that to an enzyme, a polymerase enzyme, when it's cooled, and then it's heated up again to allow those two strands to synthesize. And then what happens is it's put into this cycling, and it basically cycles over a repeated number of times to amplify the number of DNA strands. And that it's exponential. So every time it doubles, it doubles, it doubles, it doubles, it doubles, it doubles. So you're creating a huge number of cycles with a huge number of DNA. The thought is, and I say it's a thought because it's not consistent and it's not proven, but the thought is if your SARS-CoV-2 virus shows up somewhere between about 15 and 25, that means you have an active case of COVID, and most likely you have symptoms, you have viral load, you're showing all of that, you're contagious. Once you get above 30, at that point you can still show, so the first time that the DNA for SARS-CoV-2 shows up, if it's 30 to 35 or higher, that means you're amplifying, you're basically creating lots and lots and lots and lots of cycles and lots of duplications of this DNA. And at that point, it's not clear or it's unlikely that the virus is actually active, because you've had to replicate these samples, these DNA samples, so much to even see that there is a DNA sample, and it sometimes isn't the whole DNA, it's partial DNA. And once you go up above that, you literally are just replicating, and at a certain point you can find pretty much anything in that DNA sample, because you've replicated the very, very smallest segment of a DNA strand. Just something that I read this week, which I found amazing, is at 35 cycles, you have about 1 million copies of the targeted DNA. At 40 cycles, so that's 35 to 40 cycles. At 40 cycles, you have 1 trillion copies of that DNA. That's how you can see that there's a huge replication cycle happening between those two. And so what we're seeing a lot, and I'll get into this a little later, what we're seeing a lot is that the PCR tests are not having a cutoff. And they have a cutoff, but it may be a too high cutoff. So a lot of people that are not, that don't have SARS-CoV-2 or that may have had it in the past are showing up positive when they're actually not positive. So to get into this cycle threshold, this is this replication, how much amplification do you have in order to get the PCR to show up? And it's really a relative method. So one of the questions, I have 2 interns working on this that are amazing. And one of the questions I had for them is, okay, so can we have a cutoff of 30 or 35, and that will really show the answer? And the answer is no, because each specimen has a different target load, has a different load of virus in the start of it. And so we can have numbers in these thresholds that are sort of relative numbers, and we believe that somewhere between 30 and 35 should be the cutoff for an active sample. Somewhere above 35, or if you're above 35, that shows something that is not active. They're calling it asymptomatic. I actually believe that those asymptomatic people are not symptomatic. They're not passing on the viral load. They've either had it in the past and it's showing up, or they're for some reason they've had a similar coronavirus that's showing up that just had a similar DNA structure to it. So as I said, tests that register positive after 10 to 15 cycles have much more viral load than tests that are at 35 cycles. And that is sort of the contagious piece of it. So if you're contagious and you have a PCR test and it shows up at a cycle somewhere between 15 and 30, 35, you're very likely to have COVID at that point. If you are either below that or above that and you don't have symptoms, it's unlikely you have COVID, but you may still show up as being positive. This is a good slide. It's a little bit it's a lot of data, but it actually shows you some interesting stuff. So the brown bars that are tall, that sort of are a bell-shaped curve, those are the positivity samples at 1 week. So if you were positive and you had a high viral load, it shows that you're positive at 1 week. At 2 weeks, it degrades, and it's the yellow piece, so it's very, very low. And the cycle threshold, sorry, I should have said that. The cycle threshold is across the bottom. So you can see at week 1, the cycle threshold, as I said, between about 15 and 30, you're really showing that viral load. And so that is very, very accurate. At week 2, you've gone down. And at week 3, what's interesting, that's the gray line going up. At week 3, you are not getting any viral load other than at very, very high cycle thresholds. So this kind of, for those that are visual learners, this shows you that sort of difference between when you have a high viral load, where the cycle threshold is, and when you have a lower viral load, what the cycle threshold is. False positives and negatives. So one of the things that people ask me is, well, if I test positive with a PCR test, what should I do? And my answer is immediately go get yourself retested. It is likely that you have a false positive, because and let me re-clarify that. If you have all the symptoms, so if you lose your taste, if you feel like crap, you've got the flu, you know, those kind of symptoms, then if you have a positive result, it's most likely that you have COVID. If you don't have any of the symptoms, or you have very few symptoms, or you think it's allergies, for example, my recommendation is to go get another test, because the false positives are very, very high, and they get much higher when you are asymptomatic. So if you're symptomatic, the likelihood of false positives is low. If you're asymptomatic, the likelihood of false positives is extremely high. I tell a story. Elon Musk tweeted that in one day, and he wasn't using the PCR test because that takes hours to do. He was using an antigen test, which actually has much lower rates of being valid. So there are many more false positives and false negatives in antigen tests. But he went to get tested, and the first test he had, he was positive. And he said, oh, okay, you know, can I get another test just to make sure that I'm positive? The second test came back as negative. So I'm pretty sure at that point he said to the nurse, oh, well, can I get another test just to see whether it comes back positive or negative, so I know whether I am positive or negative? It came back positive. Fourth test he had done came back negative. So within a span of about an hour, same nurse, same test, same day, same hour, he had 4 tests. 2 of them were negative, 2 of them were positive. Basically told you nothing. And so, you know, that's just an example of these tests just are not conclusive. On everything they say, you know, use the test along with your symptoms, along with a whole boatload of other details with your doctor to determine if you are COVID positive or not. So why do false positives happen? False positives and false negatives happen for very different reasons. It's really important to understand the differences there, because a false positive, the reasons you'll see are much more a reason to get another test, whereas false negatives don't necessarily mean you should get another test if you think you're, you know, if you've gotten a negative and think you're positive. It's likely that your false negatives are not because of that. So false positive results may arise from a too high cycle threshold. As I said, you know, if you've got a viral load, you want to be at about somewhere between 15 and 30 for your cycle threshold. If you go above that, you're likely not to have COVID. And so that's one reason. Cross-contamination during the specimen handling or preparation. So that's whoever took the sample, mixed it with a different sample or had something on their gloves. If it was a nurse or somebody taking your test may have had, you know, a sample, some specimen on their gloves from the previous person. Cross-contamination between patient samples. So, you know, some mix up in the lab, specimen mix up, or RNA contamination during product handling. So again, these are all reasons for that your specimen has been contaminated in some other way. And for that reason, and the too high cycle threshold. For that reason, you want to get another test just to make sure. False negative results arise more often than not from improper sample collection. So for example, that you've spat into a test tube and you didn't do it correctly. You didn't put in the mixture that you're supposed to put in afterwards, or the nasal swab didn't get the right amount of your mucus. Degradation of the viral RNA during shipping or storage. So viral RNA does degrade over time, and so if it hasn't been shipped or stored correctly, or if it's been, if it was a weekend while your sample was shipped, it may have degraded by that time. Using unauthorized extraction or assay reagents. So this is for the lab doing it incorrectly. The presence of RT-PCR inhibitors, so something that stopped it from doing the process that needed to be done, mutation in the SARS-CoV virus, or failure to follow instructions for use. So those are all the reasons for false negatives. My answer is, if you get a positive and you believe you don't have any symptoms and you don't believe you have COVID, but you, for example, had to go get a test because your employer or your school or somebody required you to, you traveled. If you get a positive test, go get another one. If you get a negative result and you have all the symptoms, then you may also want to get one just to make sure that it wasn't a false negative. But those are the reasons. I think one of the interesting things with the cycle threshold is that, for example, in Florida, they passed I think it was an executive order that the governor put out, that all PCR tests, when you get the results, you have the cycle threshold there. So you can tell what your cycle threshold was for that test. And so therefore you can see whether, it was at 40, well, it was 40 and I got a positive test, then it means that I'm likely need to go get a new one. If I was 40 and I didn't have, if I was at 20 and I didn't have any of the symptoms, I may still want to get a positive another test, but at 20, it's more likely that it's because you had the viral load than not. So how do you, big question I get is, how do I refuse COVID testing? So we're seeing more and more COVID testing, especially here in California, where COVID testing is part of the reopening plan. So in order for us to go up our tiers from purple to yellow, there is no green because we're never going back to normal, as our governor told us. In order to go up each level, there has to be a certain amount of testing done in your county. And in order for the testing to be done and counted, you have to do more than the average amount of testing, which means the more testing that's done, the more testing that's needed to get into the next tier. And if you don't do enough testing in your county, then you are not allowed to progress to the next tier. You get a black mark against you for doing that. So counties are really pushing testing. It's part of why I believe for reopening of schools, they're pushing testing, because once you reopen a school, you've got 2,000 kids right there doing testing once a week, and that adds to your numbers of testing. People say, how do you refuse testing if I have to do it for work, if I have to do it to travel? My answer is, it is very difficult to refuse COVID testing. So you can try to just say no, and that's the best way, but most of the time the answer is, well, then I'm afraid you can't do what you want to do. You can't come on campus, you can't go travel, you can't do whatever. And the reason for that is that there is precedence. For example, let's say take teachers. Teachers have to have a TB test before they come in. They have to before they start employment. They have to be fingerprinted. They have to go through a whole load of requirements to be able to work in that job. COVID testing is similar to that. We are just asking you to do COVID testing so that you don't have to, that you're not spreading disease. Now, you can say what you want about the COVID testing and whether it's valid or not. As I've shown you, there are many times when it's probably not valid. But in order to, the precedence, the legal precedence is not there to refuse COVID testing. So You can try saying no. There are some employers that will say, oh, okay, well, if you don't want to do it this week, that's okay. Or, if you're required to do it every week, can you get to doing it every 2 weeks or every 3 weeks or every 4 weeks? Can you push it out? I will say that if you have to do it, our recommendation is to do the saliva test. So the options for the PCR test, which is used most often, is either a nasal swab. It can either be right in your nose, or it can be the one that goes all the way up basically into your brain, which is the one that was used at the very beginning very often. Now, mostly what's used, especially when it's a lot of people being tested at once, is the nasal one or the saliva test. Our recommendation is to do the saliva test. The reason for that is we believe getting stuff out of you is much better and you have much more control than somebody sticking something in you. I've been asked, do they have, there's pieces going around about the nasal swabs and whether the nasal swabs have something on them, whether that's how they're vaccinating people because they're putting it on the nasal swab. So when they test, they're putting, a vaccine in there, they're putting nanobots in there, do they have the technology to do that? They absolutely have the technology to put nanobots or nanoparticles on that swab up your nose and into you. 100% the technology is there. Johns Hopkins has done it. There are other, if you look it up, it's there. Are they doing it? I have no idea. Personally, I'm not going to take the risk of whether they're doing it or not because I'm going to request a saliva test instead. If your corporation, your school, your whatever, is not doing saliva testing, and quite a few of them are, but if they're not doing saliva testing, our recommendation is that you ask for a test not from the school. So you can go do this yourself. It does become somewhat expensive. You can get a saliva test at Costco for $120. It is, if you're doing a weekly test, if you're required to do it, teachers, I think, are required to do it every other week. It does become expensive. But you have to weigh the risks and the benefits of doing that. For me, doing saliva testing outweighs the nasal test and the financial piece of it, because that's what's important to me. My bodily autonomy and my bodily integrity is really important to me. For other people, they're, well, I don't care. There's likely nothing that they're going to put up my nose, so I'm not going to do it. The other option you have is to quarantine. So, one of the options is if you don't want to do the tests, you stay home. So if you're a teacher and you don't want to go in and do the testing, you're welcome to stay home. That is an option. Actually, the schools have pushed back on the governor. The governor wanted everyone to be tested every 2 weeks. The schools have pushed back because it becomes incredibly expensive. And so I do think we'll see some pushback from corporations, from schools, saying, no, we don't want to do it because it becomes very expensive. But I do see the counties wanting it because it goes towards their numbers. So they are pushing for it. Going on to COVID, so that's PCR testing. I'm going to leave that aside. I'm going to leave what you can do about it, or where to go from here to the end. I'm going to talk now about COVID vaccines and where we are with them, because they kind of fall into a similar pattern as the PCR tests, although somewhat different as well. So switching gears, where are we with the COVID vaccines? So there are over 200 COVID-19 vaccines in clinical trials worldwide. In the US, our government has paid $7.81 billion to 6 companies to create COVID vaccines, and they've also paid them for 600 million COVID vaccine doses as of the end of last month. That's how many our government has paid for. Just a reminder, we have 328 million people in the US, so that's basically 2 doses each, basically covering most of the US there. Where are we at with these? So this was also as of, this was actually of 1/24, so a week ago, 2 weeks ago, there were 21 million first doses administered. I think we're up to about 35 million now. Biden has said that he wants at least 100 million first doses in his first 100 days. He actually increased that to 150 million in his first 100 days. So where are we with these vaccines in the US? And I am going to focus on the US because that's what I know best, but this sort of covers worldwide, because most of the vaccines that are in production, that in lead production, are the same vaccines that are in lead production here. I'm not going to talk about the Chinese vaccine or the Russian vaccine, because those I don't know enough about, and they don't apply to us here in the US. We have 2 COVID vaccines that are authorized for emergency use. Again, emergency use does not mean that they have been fully approved. It means that they're still in phase 3 clinical trials. Those phase 3 clinical trials started in the summer. So we have late summer, beginning fall. So we have about 4 or 5, some of them maybe 6 months of trial data. Just to put that in perspective, most other vaccines, the quickest vaccine that was created was the mumps vaccine. That took 4 years to go from creation to full clinical trials and full FDA approval. We have never gone at this speed, which should be extremely worrying to most people. These 2 we've never had a vaccine that's come out with emergency use. We have had vaccines that have used similar technology, for example, the H1N1, the flu vaccines, that have used similar technology. I have not found a single vaccine that was rushed to market that has not failed or has not had incredibly bad consequences on the human population. And so the fact that we are rushing these out in 6 months, the fact that they only have emergency use authorization, is incredibly worrying to me. It may be that we have these vaccines and they are amazing vaccines, but the fact is that right now we don't have enough information to say either way. Nobody can sit here and say that these are safe and effective vaccines, because we do not have the data on them. One of the things that I say to people is, if you are thinking about taking the vaccine right now, the COVID-19 vaccine, any of them right now, one of the questions I ask you is, are you willing to be part of the clinical trials? Were you willing to be part of the clinical trials when it was? And if the answer is yes to that, then I say, okay, go do your research on these vaccines. If the answer is no way, I would not be part of clinical trials for vaccines, and once I get into the actual vaccines, you'll see, more about it. But if the answer is no to that, you really should not be taking the vaccines at this point. You should be waiting at least 12 months, 24 months to see what the full safety and efficacy of these vaccines is. I have on here the VSafe data, no, the VSafe marker, and this is just to show you that these emergency use vaccinations, if you're getting them today, you are still part of that clinical trial. So you're asked to sign up for VSafe. It is a text messaging system that you get texted once a day for the first week, then once a week for the next few months, then once every month, and then once every few months. And basically what they're doing is they're checking in to understand if you've had any issues, if you've had any reactions, if you've had any new symptoms, if you've had any autoimmune disorders or other things. They're checking in to understand what your reactions are. And so that just shows you that no other vaccine has that. If you go get the measles, mumps, rubella vaccine, you don't sign up for a tracker that says, yes, I'm feeling good today. Probably should, but we don't do that because they are not in this emergency use clinical trial phase. The next few slides I'm going to go through are going through the 6 vaccines that have been basically paid for by the US government and where they are in their stages, what kind of vaccines they are, and things to just think about if you are thinking of getting a vaccine. I know many on here probably are saying, I'd never get the vaccine, but many of us have family members that are considering, especially elderly family members that are considering getting the vaccine. And so while you may be solid in your perspective on the vaccine, having this knowledge is good to have so that you can have educated conversations with either family members or people that you know that are considering getting the vaccine, or that may come to you because they know that you know something about vaccines. So the 2 that we're going to talk about on this slide are the Pfizer-BioNTech and the Moderna vaccine. These are both in phase 3 clinical trials. They have both been emergency use authorized here in the US. They have also been authorized, so the Pfizer-BioNTech has been approved in 10 countries. My understanding is that those countries don't have something like emergency use authorization. They go to full approval, and they have been emergency use authorized in 22 countries, the Moderna vaccine in 5 countries and 6 countries, respectively. I put Sanofi up here because Sanofi, you'll see on the last slide of the vaccine part of this, or when I go through the vaccines, they were manufacturing, or they were creating their own vaccine, but it has not had good trials. And so they've actually shifted in Europe to helping Pfizer-BioNTech produce their vaccine. And so they now have a relationship in doing that, so that they can create more vaccine doses at a much quicker rate. So these two vaccines, the Pfizer-BioNTech and the Moderna vaccine, they are what's called mRNA vaccines, messenger RNA vaccines, and they are nucleic acid vaccines that contain genetic material, either RNA or DNA, and they carry those instructions for producing a spike protein, which is made within your cells. The key thing here is they replicate. So they put something in your cells that then causes replication. And that's one thing that my concern with these vaccines, particularly these messenger RNA vaccines, is they use replication. The two places where I believe we have replication in our bodies, number one, things like cancer. So we have no idea whether these cause cancer long-term. That's one replication process that I would be very, very concerned with. The other replication process, and we'll get into this, so interestingly, none of these clinical trials were done among pregnant women. There were 23, I believe, pregnant women, women that got pregnant after they had the vaccine, but they were not clinically studied. But yet the vaccine under emergency use authorization is available for pregnant women. And the concern there, again, your fetus is replicating cells. And so what does that do when you have this other, messenger RNA that is also replicating cells? The answer is we have no idea, because the clinical trials have not been done and pregnant women were not included in the clinical trials. These vaccines are touted to be 95% effective in preventing COVID-19. I will go into what that actually means in a minute. And then the other thing I put on these slides is the concern with these. So one of the concerns with these is people are having anaphylactic shock, and the thought is that the polyethylene glycol, the PEG, is what's causing that. PEG is an antifreeze, polyethylene glycol is an antifreeze, which is put in these vaccines because both of these vaccines have to be kept at extremely low temperatures. So the Pfizer-BioNTech has to be kept at -70 degrees. And so in order to do that, they have to put antifreeze in so the chemicals don't freeze. The other thing that I will get into, because it is important to some people, is whether they're tested or whether they're created using fetal cells. So these are not created. So in the creation of the actual vaccine, there is no fetal cells, but they are tested on fetal cells. So in the testing of whether they work, whether they create antibodies, fetal cells are used there. So what does 95% efficacy mean? Sounds wonderful. It sounds, wow, these vaccines, they do better than most other vaccines, getting 95% stopping COVID. So what does that actually mean? So after 2 doses are given, so this was the clinical trials, and this was after 2 months of the clinical trials. So there were about 21,000, 22,000 people that were given both doses, either the vaccine or the placebo. And I will say, I don't know what the placebo. So as a side note, the Pfizer-BioNTech placebo, we don't know what that was. The Moderna placebo was a saline solution, which is unusual for vaccines. Most vaccines are tested against another vaccine. The AstraZeneca vaccine, which we'll get into on the next slide, it was tested against a meningitis vaccine. So you're not doing a true randomized placebo. The Pfizer-BioNTech, it was saline in phases 1 and in clinical trial phase 1 and 2. We don't know what it was in phase 3. Some people are saying it's saline, but there's no paperwork showing exactly what it was. I've contacted Pfizer and they have not responded to me. So out of these 22,000 people, after 7 days after the dose, so it's not in the first 7 days after the second dose, but beyond that, the 2 months beyond that, 8 people got COVID who got the vaccine, and 162 people got COVID that got the placebo. So the way they're calculating the 95% is you add those two up, that's 170. So 162 people got COVID in the placebo, so therefore it's 95% effective. Only 5% got COVID in the COVID vaccine group. Sounds fabulous, really does. But when you actually look at the numbers, only 8 people out of 22,000 who got the vaccine got COVID. So that's 0.3%. Out of the placebo, out of the 22,000 that got the placebo, only 162 got COVID, which is, again, less than 1%. So I personally, if I've got my odds, yes, it's slightly more likely that I won't get COVID with the vaccine, but I still have a less than 1% chance of getting COVID with the placebo. I'll take the placebo. and I don't think those numbers are correct either, because the key here is this is after 2 months worth of data. Most of those 22,000 people have never been exposed to COVID. So we really don't know what the efficacy is. That's why you have two-year-long trials, phase 3 trials, is because you want to know that efficacy data. These numbers are going to have changed right now because we're 2 months later than when this data was shared. So more people out of the 22,000 will have been exposed to COVID. The question is, after say 12 months, how many of those 22,000 people have been exposed and how many have gotten COVID? One interesting thing I should say with all of these vaccines is that the transmission is not tested at all. So while the key metric is this metric, did you get COVID or not? Whether you may have no symptoms at all and you may not have gotten COVID, so to speak, but you may still have had the disease in your throat, and you may have coughed on someone and they have gotten COVID. Now, the interesting thing here is that that's the reason why you're told, even if you get the vaccine, you still need to wear a mask. You still need to social distance. So if you're still wearing a mask, you're still social distancing, you're still changing your lifestyle completely because of COVID, what is the point of the vaccine is my question. There is a small chance that your symptoms are not as bad, but again, my feeling is that we don't have enough safety and efficacy data to show that. So the real question is, what are you doing getting this vaccine now, today? And do you need to get it today versus 6 months' time versus 12 months' time when there is actually some data out there? The next 2 vaccines that I want to talk about, these are in phase 3 clinical trials here in the US. The AstraZeneca Oxford vaccine has been approved in 1 country and emergency authorized in 19 countries. It is the second vaccine in the UK that's being used, and in much of Europe. Mostly they're using the Pfizer-BioNTech, and then the AstraZeneca vaccine, the Moderna vaccine, is more US based. The J&J vaccine has not been approved at all yet. It is likely to be approved in the US, I believe, in the next month or so. They are getting their data ready to go before the FDA and other countries to get it approved. The one difference between the J&J vaccine versus other vaccines is that it only requires 1 dose, and it does not need to be, both the AstraZeneca and the J&J vaccine do not need to be, they can be refrigerated. They don't have to be frozen at those very, very low temperatures. These 2 are recombinant adenovirus vector vaccines. They combine the target antigen, so the SARS-CoV-2 antigen from that virus, as well as a backbone from another unrelated virus. So the AstraZeneca virus that they've used as the backbone is a chimp monkey virus, and then they've sort of inserted the SARS-CoV-2 virus into that. The J&J vaccine is using a coronavirus, so a human coronavirus, and they're inserting it on top of that. They behave more like live attenuated virus. So these ones are more likely to provoke a really robust immune response, but they also have the higher likelihood of creating actual symptoms, so severe symptoms. So you would get sort of COVID symptoms when you get these vaccines. They're not replicative, so unlike the 2 mRNA vaccines, they're not replicating in your body. Because they're based on current coronavirus vectors, they are less effective. So these are only 60 to 70% effective. The reason for that is if you've ever been exposed, and for the monkey virus, you're not likely to have been exposed to that before. But for example, for the J&J virus, if they're using a coronavirus that you've actually experienced before, you will have already created antibodies towards that. And so the vaccine you're not creating the antibodies to the SARS-CoV-2 part of that. The one thing I should say is the J&J vaccine is the only vaccine, or not the only, but of the 4 that we've talked about, is the only vaccine that is using existing technology. So the other 3 vaccines, the AstraZeneca, the Moderna, and the Pfizer vaccine, those are technologies that have never, ever been used on humans and have been approved by the FDA. So it is a true experiment as to whether these things work, how safe they are, what their long-term efficacy, long-term safety is. The concern with these 2, the ingredient that is of concern is polysorbate 80, and these are used, actually created on fetal cells and then also tested on fetal cells. I'm going to speed up a little bit here because I know we don't have too much more time left. The next two are in phase 3 and phase 2 clinical trials. The Novavax vaccine, and the Sanofi vaccine, as I mentioned, the Sanofi vaccine, they have sort of put that on the back burner because they're helping Pfizer-BioNTech with their manufacturing, and because theirs wasn't as good. These are protein-based vaccines. They can consist of the full-length spike protein or just a part of it, and they bind to the ACE2 receptor on the surface of a cell. They do create antigenic drift, which is basically if they create random mutations that alter the protein. So they try and stay on target, but there's a likelihood that they don't, and they alter it, and so therefore they don't really create antibodies towards the actual antigen that you want them to. The Novavax vaccine is also based on a technology that's currently on the market, the flu block vaccine. You know, I say to people that at least gives us some peace of mind. I still have issues with a lot of the vaccines, so it doesn't really give me peace of mind, but it at least has gone through, the technology has gone through full clinical trials and been approved by the FDA, so that, you know, it at least says it's not completely an experiment, these ones. The concern here is this Quillaja saponis, which is a soapbox tree. It's an adjuvant. An adjuvant stimulates the immune system. It also, you know, hyperstimulates your immune system. That's what it's supposed to do for the vaccine. The question is, what else does it hyperstimulate? So it can hyperstimulate, for example, the autoimmune system. And these 2 are tested on fetal cells. They're not created with fetal cells. So then the question becomes, how do you decide whether you take a vaccine? And this is the truth for any vaccine or any product that you're considering taking into your body, even food. You really want to do a risk to benefit analysis. This risk to benefit analysis is very personal. So my risk to benefit analysis is completely different from a 90-year-old person that's autoimmune, you know, has autoimmune disorders, is on 10 medications, versus a child that is, you know, 5 years old and is healthy and eating organic food. Those 3 have very, very different risk to benefit analyses, especially for COVID, because we know that COVID is much more virulent and likely to cause much greater effects on elderly people versus young people. The one thing I will say, the key piece of this is you can always vaccinate. You can never unvaccinate yourself. You can never get all of that out. When you get injected with an mRNA vaccine that is replicating, or any other vaccine, you know, the ingredients or whatever else, those are in your body. You don't get to get those out. There's no way of extracting them out of your body. So this is a really serious consideration, because it's not something you can undo. You can undo some of it by detoxing, but you really can never undo the full scale of the vaccination. So you need to look at the risk and the benefit. So the risk of the disease, sort of on the left-hand side here, I have the risk of the disease, and the right-hand side, the risk of the vaccine, or what you need to take into consideration. So the survival rates for COVID, we know, are extremely high. Even among people above 70 years old, they are about 95%. So most people that get COVID, that get SARS-CoV-2, are going to survive. We know that there are more and more COVID treatments. So, you know, what is the likelihood that you survive? You can be treated and you have, you know, you can live your life. There are people who have long-term health effects. We have to realize that, whether it's something that's, you know, considered mundane, like losing your smell and your taste, which for people who that happens to is not mundane, or to, you know, internal organ issues. So there are definitely long-term health effects on it, and that should be a consideration. The other question that we don't have an answer to is, how long does immunity last? So if you had COVID, how long does it last? Is your immunity there for a year? Is it there for 2 years? Is it there for 5 years? It really depends on the strains and the mutation of the virus. But you know, and do you have immunity to variants? If you had COVID one variant, are you immune to the other variant? We don't know. Those are questions that we don't know yet. On the other piece of this, the risk of the vaccine and adverse reaction. So what's the likelihood of an adverse reaction? I'll go into that a little bit. We don't know whether it causes cancer, whether they cause autoimmune disorders, whether they cause infertility, fetal issues, mutagenicity. We have no idea because these vaccines that are currently on the market have never been tested. So you have no idea of the long-term effects on your body. Antibody-dependent enhancement, I'll go into that quickly, is an issue. We don't know how long immunity lasts for these. We've been told that there may be boosters. For example, Moderna is coming up with a vaccine number 3. I worry about that significantly because the side effects for the dose number 2 are huge. Means the side effects for dose number 3 are also likely to be much greater. And we haven't, as I said, we haven't evaluated whether they stop transmission. You still have to wear a mask, you still have to social distance. I'm going to go into 2 pieces of this. I'm going to hurry along here. Antibody-dependent enhancement. So what this is basically what they showed with the SARS coronavirus vaccines that have been in development for 10, 15 years since MERS and SARS were here, is what they found was in the preclinical trials, the animal studies, none of the vaccines that are currently on the market went through full animal studies. But what they found was they gave the vaccine to the animals, they created amazing antibodies, looks like the vaccine was doing great. And then when they exposed the animals to the virus, all of those animals, or 95% of them, died. Or had, you know, the rest of the 5% had really bad reactions. And that's through this antibody enhancement. And what it means is that basically, when you get the vaccine, it needs to stimulate neutralizing antibodies as opposed to non-neutralizing antibodies. Non-neutralizing antibodies basically says that there's some form of binding of the antibody to the virus, but it doesn't neutralize it, doesn't get rid of it. And when you have non-neutralizing antibodies, you basically can create a really severe hyperinflammatory response. So basically, the question then is, if you have the COVID vaccine and you're exposed to COVID, what is the likelihood of this antibody dependent enhancement? What's the likelihood of you having this hyperinflammatory response? We don't know because not enough people. We saw only 170 people got COVID after the vaccine. We really don't know. And then the other question is, if you've had the first dose of the vaccine and you get COVID, as I said, does that happen? But the other question is if you've had COVID-19, the actual disease, and you get the vaccine, does that have that effect as well? We really don't know. So again, just to be clear, none of the trials were done on people that had had COVID prior to getting the vaccine. But yet they're telling people if you had COVID over 3 months ago, go ahead and get the vaccine. No clinical trials again. What you may be getting from this talk is there really are no clinical trials that have investigated any of this. The true measure in the clinical trials is one and only purpose: do you get COVID vaccine? Do you get COVID after getting the COVID vaccine? The adverse events reporting system, I'm going to rush through this a little bit, but if you go to VAERS, that's where if you have an adverse reaction to any vaccine, it should be reported to the VAERS system. There was a study done by Harvard which showed that fewer than 1% of adverse events are actually reported. So these numbers that I'm showing you may be many, many more times that, because not all of them are reported in there. I do think the COVID vaccines likely have a higher reporting because they're emergency use and because of the vSafe texting app that sort of stays on top of you. But what we saw in the first 2 months of the vaccine, this is I think about 2 months worth of data, is that there were a total of 9,000 reports of adverse events. Most of them were non-serious, but non-serious still means that they put you out for a day or two that you couldn't do your normal activities. So, to me, that's a serious adverse event. To VAERS, that's not a serious adverse event. Serious adverse events, there were 11%, there are about 1,000 of them. And those include deaths. I think up to today, there are around 1,000 deaths after the vaccine. Now, the tough thing with the deaths is that because the vaccine is being given to mostly elderly people, most of those are being written off. So, you know, oh, they were 89 years old. You know, people, if you read the stories, they were fine before the vaccine and then they died. But if you read the VAERS reports, they're being written off because, oh, they were 89 years old. Well, they would, and in a nursing home, they would have died anyway in the next, you know, few days or few months anyway. So we're not really seeing the true numbers there or what is going on there. One other piece of information is that there is no liability. The PrEP Act, which is the Preparedness Act, basically allows says there is no liability. So it doesn't matter whether you die from these or not. There's nothing you can do about it. There is a compensation program you can go to, but good luck there. Vaccine mandates, what do I see? I don't see federal and state vaccine mandates in general. I do see them on a maybe smaller level, for example, for healthcare workers or for teachers, those types of things. I do see societal mandates. So I have a picture of people with masks. I think we're going in exactly the same direction. You're not going to be able to get in a store. You're not going to be able to travel without the vaccines. I think these changes, unless we stand up, I think these changes are here to stay, like after 9/11, the TSA changes. And how do you refuse the COVID vaccine? Just say no. On this one, you have to say no. Because there are numbers that are saying no. So we see 50, 60% of healthcare workers in some places saying no. You can say no. The other thing that I will say is get out of the system. If you are being told you have to get a vaccine because you're a teacher, get out of the system, go teach somewhere else, become a tutor, do something else you love. And I know that sounds harsh, but it is the reality of today. And I really think we need to stand up for that. You can get medical exemptions or religious belief exemptions as well. But if you do that, you have to remember you're staying in the system. Let me just see where we're at. Okay, and this is my last slide. Where do I see the future of COVID? I actually think we're going to see COVID disappear relatively quickly in this next 6 months. I think the reason for it is not the reason we're going to be given. I think we're going to be told that the vaccine is doing wonders. I think the real reason is that the symptoms or the COVID vaccine positive symptom definition is going to be changed to be stricter. So fewer people are going to be diagnosed with COVID. I think we're going to see deaths with COVID reported less often. So right now, or the last year, if you had a heart attack and you had COVID, you were a COVID death. I think we're going to see more heart attack deaths. And as I said, I think the PCR cycle is going to be brought below 35. So all of those false positives that we've seen that have not been counted as false positives are going to go away. As I said, I don't think any of this is going to be acknowledged. I think it's all going to be the vaccine is wonderful, and that's is what's going to have saved us. What do you need to do? You need to research, you need to speak, join your local health freedom groups, and just say no and get out of the system. So I went, I think, not quite over, but I know I rushed through a lot of that. It's a lot of information. I would say, you know, feel free to go through it again. I know these are going to be, these are recorded and you can go through it again. I also recommend joining our email list, A Voice for Choice Advocacy, and you're welcome to also email me, Christina at A Voice for Choice Advocacy, or info at A Voice for Choice Advocacy, and I will get back to you with any questions you have.